Welcome to Life in Biology. I'm Dr. Joel Graff, and today we're going to be talking about what it takes to be human. That is, how many genes does it take to come up with the complexity of what ends up being a human body and human mind and human everything. So the first place we're going to start is with the central dogma of biology, if you can believe it. Let me rearrange this at the end a little bit. Okay, so how to make the most of what you have. When the human genome was being sequenced, the DNA sequence of the human genome was being sequenced, and a draft came out in the early 2000s, there were people that were guessing how many genes would be in the human genome, and the total number ended up being between 20 and 25,000 protein coding genes in the human genome. And this was much lower than nearly everyone's guess had been. And so we're going to talk about how you can start with DNA and have 20,000 genes, but how that can become 100,000 different mRNAs. And we know that mRNAs encode for proteins. So how do we end up with over 1 million different protein variations from those numbers there? Doesn't seem to make much sense, but there's a couple major reasons why we have so much variability in our ability to make proteins. So when DNA gets transcribed to RNA, there is a little bit of a detour because the DNA is protein coding information is spaced out along the genome so that you could draw it as if it were boxes would be the protein coding sequence and have them separated out. And this isn't really drawn to scale. These lines between the boxes would be very long, and you would have the information for your protein is scattered across a very large distance for most genes in the human genome. So if you transcribe the DNA and you make a pre-mRNA molecule, that pre-mRNA molecule might be tens of thousands of base pair long and end up with a final mRNA product that is 1 or 2 or 3,000 base pairs long, or nucleotides long, excuse me. It's single-stranded, so we don't say base pair. So how do we end up with 100,000 different RNAs? Well, I numbered the boxes here showing the protein coding sequence. Hi, Lauren. And we have the boxes are going to, if they're going to be made into the final RNA, they're called exons, and I've labeled them 1, 2, and 3. As you go from pre-mRNA to the messenger RNA, splicing occurs, and I've drawn here that 1, 2, and 3 could be used to make an RNA, and there's another arrow here showing that 2 was left out in this case, and that's called alternative splicing. We'll talk about that more in a second. And then once you have the protein, those proteins can be modified, and that's how you can end up with about 1 million different modified protein versions. So let's start with talking about alternative splicing first. So I'm giving an example of a gene called TRIM5 alpha. This is a protein coding gene that the TRIM5 alpha is the largest protein that can be coded by this gene, and it has anti-HIV properties. If you look at the genome arrangement for this gene, there are multiple exons scattered along a very long distance of the genome, and here the exons are numbered 2 through 8. I don't know what happened to exon 1, but when I looked it up online, there didn't seem to be one. But if you transcribe this into an RNA, and then splice out all the intron sequence between each of the exons, you can end up with a shorter RNA, and where exon 1, 2, 3, 4, 5, 6, 7, and 8 are all connected together, and we lost all of that intronic sequence. And then what can happen is that mRNA, this is a 5' untranslated region, and here's a 3' untranslated region, that mRNA can be read by a ribosome and made into the TRIM5 alpha protein. If we take this TRIM5 gene and we only stick the first 3 exons together, we can make an mRNA out of that. So here I've got 1, 2, and 3, and this is a different RNA variant. So if the RNAs are different, they're called variants. And this is called trim-I. So trim-A was the one that coded for trim-5 alpha. Trim-5I will code for trim-5 iota. And so I've got the proteins drawn here, and you can see that trim-5 protein, I've got it drawn as two kind of lobes. And what one lobe does is it acts as an E3 ubiquitin ligase. I'll talk about that in other videos, and I've talked about it before. And then the green part of the protein here interacts with the HIV molecule. And so between those two, you have a functional molecule that can go find HIV and put ubiquitins on it. However, if you don't include that information from the 3' end of the gene, and you're only working with exons 2, 3, and 4, the protein is only going to be the purple part. It will not have that green part. And so this would be a protein that would be unable to attack and counter HIV. So if your cells decide to switch from going from trim-5A to trim-5I, and you go from making trim-5 alpha protein to trim-5 iota protein, those are called protein isoforms, then your ability to block HIV viral replication is severely diminished. So that was alternative splicing. And then the other mechanism, going from 100,000 different mRNAs to having over 1 million different protein variations, is done through post-translational modification. And again, I'm not a very good artist, and so this squiggly line going all over the place is supposed to represent some random protein. And then in purple, I've drawn on different functional groups onto this protein. And so I'm going to start here, and we'll work our way around in this way. Here, what I did is I drew, there's an oxygen, a phosphorus, and then a bunch of other oxygens. That's called a phosphate group. And so proteins can be phosphorylated. So you can have the unphosphorylated version of a protein or the phosphorylated version of a protein. Somewhere else in the protein might be modified with the addition of a different type of functional group, and in this case it would be a methyl group. We learned about a methyl group is a carbon with 3 hydrogens on it connected to whatever it's modifying. So the methyl group there. Over here I've stuck a ubiquitin on. You can see from my drawing that is a protein, a small protein, and it's been stuck onto the large protein. And so if you can actually modify proteins by sticking on another protein to that first protein. And then finally, I show glycosylation. And so these are supposed to be hexagons to represent glucose or other 6-carbon sugars. And I have them attached in a bit of a string to the protein. So each one of these modifications could change the property of whatever protein this is. And if you have phosphorylation, no methylation, no ubiquitin, and no glycosylation, that would be one version. Or it could be methylated without any of the others, or it could be phosphorylated and methylated. And so you can see there where with all these possibilities for adding modifications to a protein, post-translational modification, you can end up with proteins that have different functions. So if you want to change a protein's function, you could add in a functional group. All right, so that's how we go from having a mere 20,000 genes in the human genome, protein coding genes in the human genome, to having over 1 million different variations of proteins possible in the cell. All right, like and subscribe if you want, or not.